Okay, students, this is April 4th, and I had given my fourth and fifth lecture to you the other day, and I was feeling a little guilty because I didn't have my hands on some of my transparencies. And some of these transparencies are so good because they make things real clear, or clearer. So I wanted to go over them with you. If you look at the lateral view of the left hemisphere, of the brain, you're going to be able to see some of the structures more clearly that I had discussed in lectures 4 and 5. So this is a little bit of an addendum and a review. And I'll give you this review, and then later on, and hopefully later on this afternoon, I'll do lecture 6 for you. But if you look at the left hemisphere of the cerebrum, here's the cerebrum, the cerebellum, and here's the brainstem. Right here is the central sulcus. And I can see that on the monitor. There's the central sulcus, and it divides the frontal lobe from the parietal lobe. It's also an important landmark because the gyrus, remember, right in front of the central sulcus is the precentral gyrus, and that gyrus, this tissue all in here, is involved in gross and fine motor movements. Whenever you want to make a movement throughout your body, utilizing the skeletal muscles, those muscles that you have somatic control over, it is going to come from this tissue right in front of the central sulcus. It's a general motor area, and I wanted that to be more defined. Now, right behind that is the post-central gyrus. And in the post-central gyrus, right after the central sulcus, all of that is tactile sense. And I wanted that to be real clear. And in the other diagram that I showed you on the transparency, it wasn't as clear. I know it is in your textbook, but I wanted to point that out. Of course, the parietal lobes on both sides are involved in general tactile senses, how your body feels as far as the outside temperature is concerned, the clothes on your body, the shoes on your feet, everything that you can feel is rendered sensible by that post-central gyrus in this area in the parietal lobes on both sides, of course. But I wanted to show you this, and this is much clearer and more definitive of that real important landmark, that central sulcus. Remember, the central sulcus is here. Here is the lateral sulcus, which separates the frontal lobe from the temporal lobe, and the frontal lobe from the parietal lobe. The transverse fissure right here separates the cerebrum from the cerebellum. So below the lateral sulcus is the temporal lobe. Above that lateral sulcus is the frontal and parietal lobes. And of course, the big general landmark is the longitudinal fissure, which pulls the brain apart into two halves or two hemispheres, which are held together again by what? The corpus callosum. Okay, good. I was worried about that. Now, this one was one I wanted to make sure I showed you, and I described it, but I didn't like the transparency, and this transparency makes it so much clearer. If you did a section, the top of the brain up, just as you would open a jewel box, you lift it up and you look inside, and here you can clearly see that 1/4 of an inch all around the brain, the cerebral cortex, of course, is illustrated in a darker color, which in the real brain is gray in color because you're seeing millions of dendrites and cell bodies which are non-myelinated. And the myelinated axons are angled towards the center of the brain, and that's why it's illustrated white in color because of the myelinated axons. Now, these darker areas that you see right here are referred to as the basal nuclei. Now, again, a nuclei in the central nervous system is a bunch of cell bodies. And of course, these cell bodies don't have myelin on their dendrites or on the cell body itself. So when you have bunches of them together, they're going to appear as gray spots within the white matter of the brain. And a nuclei has a number of terms. We talked about the nuclei in the nucleus of a cell, a plant cell or an animal cell, and that nuclei contains the DNA on the chromosomes. We use that term for nuclei. We also use it generally in the CNS as a cluster of cell bodies. So it's used then, and of course they talk about the nucleus of an atom. So there's three terms which refer, the same word refers to different structures. And the same bunch of cell bodies in the PNS are deemed a ganglia. Now, sometimes they call these the basal ganglia, but that's not totally accurate. But calling it a basal nuclei is accurate. Now, another word, the darkened area all around the basal nuclei is referred to as the substantia nigra, which means a larger darkened area. And it's the substantia nigra that the basal nuclei are located in. And the basal nuclei, if you remember, releases a neurotransmitter known as dopamine. Dopamine is a very important neurotransmitter because what it does is it controls the secretion of acetylcholine. So there's a proper amount of acetylcholine released. When acetylcholine is not properly accounted for, if too much or too little acetylcholine is released, then there are drastic problems such as Parkinson's disease. But I did not have this transparency, and pictures make thousands of words, don't they? And these basal nuclei are so much clearer on this transparency, and I love it. And I didn't have it the other day. So there is what they look like. If I had you in class, I would run off 20 of these, and you'd have each one of you would have one of these so you could refer to and look at the basal nuclei because they make so much more sense when you actually see them on the transparency. Okay. Now, the next thing I wanted to talk about was the discussion of the cerebral spinal fluid. And I think that isn't the one I want to show you. It's this other one. The cerebral spinal fluid is that very special fluid that is made by the choroid plexus, which is a bunch of capillaries inside each one of the ventricles. And as the other day, I remember telling you, we had 4 ventricles in the brain, and they're just spaces in the brain filled with the cerebral spinal fluid. Now, these spaces aren't real large, but they certainly do exist. And they're not real large because we don't have a tremendous amount of cerebral spinal fluid in the average brain and spinal cord. Remember, anywhere from 4 to 6, maybe 8 ounces of cerebral spinal fluid, which is constantly being made by the choroid plexus and extracted from the plasma of the blood by the ependymal cells, which are neuroglia. And the choroid plexus is that bunch of capillaries which releases it. This is a good diagram because it shows one of the lateral ventricles here. And this ventricle right here is the third ventricle. Remember, the third ventricle is inside the thalamus, and this is the fourth ventricle. And it just is real clear in this little diagram. So this is the lateral ventricle filled with cerebral spinal fluid. It flows from the lateral ventricle through this little duct known as the interventricular foramen to the third ventricle, which the outside of it is really the thalamus, and from the third ventricle down this long cerebral aqueduct. This was the interventricular foramen. This is the cerebral aqueduct, which carries the cerebral spinal fluid to the fourth ventricle. From the fourth ventricle, there's a hole which takes the fluid all the way around the outside of the brain, the front of the brain, and down and around the entire spinal cord, as well as through a little hole in the middle of the spinal cord, which is the central canal. Now, blood vessels, the choroid plexus containing the ependymal cells, extract the fluid again from the plasma, and you have fresh fluid constantly coming in from the ependymal cells, and the old cerebral spinal fluid is being drained by the choroid, not the choroid, the arachnoid villus, and I'll describe those later, but that shows it to you a little clearer. And I've got another one. I try to show as many transparencies as possible to make things clear. Okay. Now, this is a little bit bigger. Let me turn this so you can see it clearly. In this illustration, the artist has made the ventricles real big. They're certainly not that big, but he's made these structures real large so you can see them, and colored in blue, containing fluids. So I guess that's why they're blue for water. But here is the lateral ventricle in the right hemisphere. There's the lateral ventricle in the left hemisphere. Here's the interventricular foramen draining into the third ventricle, and from the third ventricle down that long cerebral aqueduct all the way down, and then it flows around the outside of the brain and then down around again the outside of the cord. And then he's shown you right here where the fluid is draining right down the middle of the central canal of the spinal cord. So that makes it clearer too. And this one has an artist's rendering of the inside of the third ventricle, the thalamus, and they're showing you this ruffled pink structure right there. That is the choroid plexus. And there is one in the right and left ventricle, one in the third, and one down here in the fourth. And you can see how the artist has also illustrated it in that darker pink color in the fourth ventricle. That's the choroid plexus. And I wanted you to have a visual on that. And this transparency shows it pretty clearly too. And it shows the flow of the fluid. Here ventricle. The arrows are showing you the flow of the fluid to the fourth. And there is the choroid plexus in the fourth ventricle, all the way down on the outside of the cerebellum here, and then it's all the way around the outside of the brain again, and then it's showing how it flows down around the entire cord and up. And this one has one in your textbook very similar, but it's more dramatically illustrated here, showing you that choroid plexus. Remember, it has those ependymal cells that extract from the plasma, the cerebral spinal fluid, which doesn't have certain amino acids and certain hormones which could do damage to the brain. And the cerebral spinal fluid is very specially filtered so that it doesn't do damage to the brain. Another structure known as the astrocytes, we'll talk about a little later. They wrap around the capillaries that feed the brain and also protect the brain. The brain develops this protection around the end of the first year of life, and it's referred to as the blood-brain barrier in the fetal stages. And in, as I said, the first year of life, the baby doesn't have a blood-brain barrier. So you have to be very, very careful because anything that's given to the baby, medications, anything can go into the baby's brain. And that's why we're very, very careful about working with mothers and telling mothers to be very careful of medications and alcohol because their babies in the fetal stages do not have, throughout that entire gestation, do not have a blood-brain barrier. Okay. All right. That was just 16 minutes, but I feel a little bit better. I wanted to go over those illustrations with you. I have not received, but just a few of you, that you've seen all the videos up through the 5th video. And I want you to let me know, of course, in the next day or so, because I'm counting that as a quiz grade. And tell me that you are up to date on viewing the videos and doing the study guides and reading your text, and I'll feel much better. Well, I will come back later and do lecture 6 for you, but I wanted to make sure that was real clear, and that's kind of an